another story, something to get off my chest.
Hello and welcome to another Piper Pod. Today we're going to do a short one in the anatomy physiology theme, and we're going to talk about mitosis. Now, we spent time during the reproduction side of the house talking about meiosis and the way daughter cells are formed in forms of sperm and ovum. This time mitosis differs because it creates exact replicas of the cells that it's dividing, and it happens with every cell in your body that contains a nucleus can go through mitosis. So the way we grow, develop, change, replace ourselves is through stages of mitosis. So this is happening in our bodies trillions of times each day, and this will just go through the process of how the whole thing works. So when we look at mitosis, the main thing that people focus on, and the main aspect that is focused on, is DNA replication. Now, I'm not going to go through the absolute ins and out of protein synthesis in this lesson. That's going to be a different lesson because it differs slightly in the process. When we talk about DNA replication, we're talking about the interphase side of the house where the cell is planning to duplicate itself. Now, deoxyribonucleic acid, we all know, is the backbone or the building blocks for everything that makes us us. It contains all of our genetic material that is used in order to create our structure and a blueprint of how to design our body. It tells us what sort of enzymes we carry more of, what sort of features we're going to show, and gives us the probability of what we can pass on to our next generation. Now, when we look at a DNA structure, the main things to focus on is the DNA is broken up into based nucleic pairs. That means we have 4 different nucleotides within DNA. That's it. So everything on the planet that contains DNA, so everything that is alive, plants, animals, microbes, humans, we all have the same 4 based nucleic pairs that make up our DNA. And it's literally that structure and that formation or combination that makes us different. Now, the 4 based nucleic pairs are adenine, thymine, guanine, and cytosine, and they branch together. So adenine will bind with thymine and guanine will bind with cytosine, and vice versa. So when they're replicating themselves, those two are attracted to one another. Now, the only example where this differs is when we start talking about messenger RNA, which we will in a different lesson, and not DNA. Now, in that case, uracil comes into play and it replaces thymine. So uracil will bind with adenine in order to move out of the cell. Now, if it wasn't for uracil, then we wouldn't be making messenger RNA, we'd be making messenger DNA, which is essentially what we're talking about in this particular lesson. So that's the difference, and that's what DNA is made up of. Now, all your DNA within your cell is housed within your nucleus itself. Now, to try to appreciate how impressive that is, we have to remember when we're talking about a cell, we're talking about something that the naked eye can't see. So we're already talking about microstructures. Within that microstructure, we have a nucleus, which takes up, in some cells, around 30%, in other cells, about 1% of the actual size of the particular cell, especially when we're talking about our myocytes, even less than 1% really. And within that particular nucleus will house all of our DNA. Now, DNA is a double helix structure. Now, I know we've all heard it being called that, and we sort of understand that it's two chains, but those base nucleic pairs are formed in chains, and then they're twisted around themselves, mostly to reduce their length in size. Now, the size or the length of a DNA chain, when it's fully unraveled, if you will, it is around 2 meters in every cell. So you have 2 meters of DNA in every one of your cells. And if you were to put all your DNA together, some people theorize that that would equal 2 times the circumference of the solar system. That's how long it would be. So we're talking about extremely long structures. And now this individual 2 meters of DNA has to fit inside our cells. Now the way it does that is DNA will become, part of your DNA will turn off basically and be inactive. So the whole aspect of your DNA is not activated in use. So when you think about endocrine cells, the part of the DNA to make the particular hormone will be more active. And when you're talking about myosin cells, the process of making different muscle tissue or increasing protein for muscles to develop will be turned on, whereas the rest of it will essentially be put away. Now the whole DNA structure twists itself around this special protein called histone cores. Now, as they move around the histone cores, they obviously reduce in overall length, and they can have different areas of the DNA accessible for protein synthesis. Now, when all the DNA is wrapped around these histone cores, they're called what's known as a chromatid. So when they wrap around themselves, they become a chromatid, and then that chromatid will actually condense on itself and wrap itself up. Okay, so think of this as a Christmas light nightmare, where each individual strand of Christmas light has been wrapped around a pen, and then all of those pens have been wrapped around one another, all in one tight, messy bundle called a compact chromatin. And that's essentially how they stay in the cells full time. Now, different parts can open up at different times depending on what is needed to be developed, but that's basically how they are. Now, from there, our classic chromosome that we all hear about, chromosomes only really form themselves during active stages of mitosis. And the classic way that we see them all lined up down the cell is during a phase called metaphase of mitosis, which we'll go through. So generally speaking, DNA is wrapped up as chromatids, and they're inside the nucleus just existing, being there for the recipe for protein synthesis and development. Now when it comes time for DNA replication in an aspect known as the cell cycle, DNA replication occurs during interphase of mitosis. Now the way it works is the double helix within the actual nucleus, they start to unravel from the histones, and then the double helix starts to separate. Now this is aided by an enzyme called helicase. So helicase enzyme will actually come along and unwind the double helix into 2 different strands. Now the strands will separate, and they'll have what's known as a primary or a leading strand, and then they'll have a lagging strand. Now the leading strand and the lagging strand will actually synthesize DNA in different ways. So when we look at the leading strand, it will start at one end, and it will just bring in its base nucleic pairs, and it will build an exact replica of the lagging strand. Easy enough. So with the help of DNA polymerase, it will actually bring in base nucleic pairs, and it will replicate itself against the actual leading strand. Nice and easy. So adenine will bind with thymine, cytosine will bind with guanine, and all of a sudden you'll have what you had before with the lagging strand. It will just be a brand new DNA strand, right? The exact copy. The lagging strand, on the other hand, is a little bit more complicated. it uses processes, instead of building it in one big long chain, it will actually build it in smaller pieces. So it uses primase in order to bind RNA primers to create small segments of nucleic chains, okay, or polymerase. So when these DNA polymerases are formed, then they're bound together by DNA ligase. So instead of building it in one big long chain, it will just build it in small segments and then combine it in DNA ligase. But the end result, once it's all done and all DNA is replicated, remember this is done during the interphase of mitosis, then you will end up with double the amount of DNA within the nucleus. And from there, it starts to form itself down into individual chromatids, and then eventually becomes the chromosomes that we know before the split occurs. So let's move on. Knowing what DNA is, how it's replicated, and where it's housed in the body, we'll have a look at the different stages of mitosis and how a new cell is formed. So this cell cycle that we speak about, the majority of it, as you can see, is interphase that we spoke about, but only S phase itself during the cell cycle is where the DNA is actually duplicated. Now during G1 phase, this is where the rest of the contents of the cells are actually duplicated. So you get double Golgi apparatus, double endoplasmic reticulums, more ribosomes, more mitochondria. Everything just starts duplicating itself within its cell. Then you will duplicate the actual chromosomes, and then during G2, this is where everything just basically gets checked. So we want to make sure that the chromosomes that have been produced are exact replicas of their partner. Because if they're not, then essentially you are changing your genetic code, and if that does happen, then what happens is the cell itself, if it can't be fixed, will actually just die. Okay, and you'll actually end up with cell death as opposed to cell replication if the DNA replication isn't done perfectly. But once all the double checks and everything is done, then we'll move into the other stages of mitosis. So this phase is called interphase. It obviously takes the longest, but a lot of the time it's not included in the active phases of mitosis. The classic stages of mitosis starts with prophase. Now prophase, if we exclude interphase, is the longest phase of mitosis itself. This is where your chromatids become chromosomes, and this is where you can really see the chromosome structures within the cell itself. Even under a microscope you can actually see it. Now centraloids, which are very specialized proteins that form during mitosis in order to aid in the separation of these chromosomes, will actually start moving to different poles, and they'll extend fibers into the nucleus, which is breaking down, and connect to the will start connecting to the chromosomes itself. So at this stage you'll have all the chromosomes in what used to be the nucleus and now just starting to float around the cytoplasm, and your centraloids with their spindles starting to form. So this is like all the preparatory phases of cell division. When we move into metaphase, this is just a phase of saying that everything is pretty. Everything is lined up and everything is ready. So as you can imagine, this is the shortest phase, but when you see it under a microscope, it looks very impressive because all the chromosomes are lined up in the center of the cell. You can't really see the spindles unless you have a good microscope, but you'll see them perfectly lined up with structures on either side of the cell itself. And that is metaphase, already lined up in formation to be split. When we move into anaphase, this is where the chromosomes actually start to split into individual chromatids themselves. Okay? And they start to get pulled to opposite sides of the cell. When we move into telophase, this is where two cells start to form. This is where the chromatids start to unwind back into chromatin, and then the new nucleus is formed around them, so their new envelope is formed around them. The spindles have disappeared and been broken down, and the cell starts to pinch into itself and form two cells. All the other organelles within the cells that have separated as well will start to move into their areas, and they'll start to basically become a brand new cell. So telophase is the formation of new life, basically. Now when we move into cytokinesis, which is a cytoplasm division, this is where we have separation. Okay, so telophase is all the design of becoming an independent cell, and when we finally get to cytokinesis, this is when we have two exact cells, and each daughter cell is a replica of themselves. And obviously that means we have new aspects of life, if you want to call it that, but new development within our body, okay, a new structure. Now when we talk about cells, obviously different cells require different rates of mitosis. So not every cell will actually need to be replicated at the same time. And when we have wounds, we cut ourselves, or we tear our muscles, then mitosis picks up. In order to go through cellular regeneration and repair, this mitosis aspect will happen in order to form those new structures. Now once they get into this cytoplasm stage, like cytoplasm division, cytokinesis, then from here they can start to specialize in the different types of cell structures that we find throughout our body. Now I don't mean that every cell becomes the exact same cell like a stem cell. That means when you're looking at the skin, we obviously have different types of cells that are needed at different layers. So if a cell is divided, then it can actually change itself in order to become part of a structure with slightly different shape, different amount of organisms within them, in order to perform the function of that particular cell. Okay, nice short one, going through the stages of mitosis. Just wanted more of an introduction because I spent a long time talking on meiosis. So just to show the difference in mitosis. The next and final lesson for the anatomy physiology side of the house, we'll be looking at protein synthesis and how proteins themselves are formed in the cell and then moved out of the cell to be utilized in the body. Until next time, take care.